person. Do you know how to access the drive?
No, I don't. But that's why I asked in the beginning of the session. Where is it? Yeah, but I'm here. Okay. Okay, how do I make it?
Window, you can do window and then full screen, I guess.
Where is it?
Otherwise, you press the green thing.
Okay.
Yeah.
All right, so they say best things are worth waiting, but I hope that is true. I'm going to talk about our web-based interface called ImmuneRegulation. ImmuneRegulation aims to help investigators to interact with massive data sets on gene regulation. And in terms of gene regulators, gene expression regulators, we are specifically focused on genetic variants and transcription factors that regulate gene expression in response to stimuli, vaccines, pathogens, and therapies. And we developed ImmuneRegulation in response to domain experts in immunology project consortium that needed this kind of an interface to interactively explore massive data sets that are being developed by HIPSI. However, we also included public databases that are available on ImmuneRegulation and through a user-friendly interface. When we talk about regulators of gene expression, we specifically talk about Eqtls and transcription factors, and Eqtl is just a DNA variant that's going to alter gene expression. It may not alter gene expression, then it's not an Eqtl. And if it alters gene expression in the middle panel, then then we call it an Eqtl. And then, or maybe it doesn't alter gene expression normally, but if the cell is exposed to a stimulant, for example, a toxin or a vaccine, then it's a response Eqtl. So our database covers all these different types of data. And an Eqtl is so the variant may be affecting a gene within one cell type, but it may be affecting a different gene in a different cell type. So they are cell type and tissue specific usually. And also an Eqtl may be affecting a nearby gene, then it's called a ccQtl, or it may be affecting a gene that is further away or in a different chromosome, then we call it a trans-Eqtl. And within ImmuneRegulation, we have a central repository of gene regulation data sets, and it's currently the largest centrally integrated resource available in the literature on transcriptome regulation on whole blood and blood cell types. We have whole blood B cells, monocytes, CD4 plus T cells, etc., and different exposure types. Overall, we have information on 43,000 genotyped individuals from 50,000 more or less gene expression experiments, and overall you can query for more than 220 million Eqtls within our database based on any p-value that you're interested in. And also for our consortium, we have analysis pipelines to identify Eqtls, both cis and trans, and different flu and chikungunya and other data sets that are being developed. And in terms of transcription factors, which are proteins involved in gene regulation, gene expression regulation, we incorporated ENCODE data set from about 2,000 ChIP-seq data, and we extracted about 14 million gene expression hits from ChIP-seq peaks and integrated them within ImmuneRegulation. And we also wanted to provide the users an opportunity to look at traits associated with these variants. So we integrated the NHGRI EBI catalog of GWAS studies, and that was about more than 67,000 variants, SNPs, that are associated with specific traits. And this is also integrated fully within the interface. So we integrate multiple different types of data sets within ImmuneRegulation, and we enable interactive query and visualization. It's simple and intuitive browser interface, helps explore filtered data sets and submit and modify queries. And users can also upload private data sets and explore them within the same interface. And at the privacy of their own computer. Within the query interface, they can do Manhattan plots or p-value based plots or the transcription data sets. And we also have analysis pipelines for Eqtl data analysis. And the website has example queries, tutorial slides, FAQ section, and feedback form. And feel free to look at our website and also provide us feedback. Anything that you want us to be changed, we will change them. And it runs on all modern web browsers. No external plugins are needed. cis-Eqtl datasets are compressed and indexed using TabX. Trans-Eqtl datasets and the GWAS catalog datasets are stored in tab-limited text files. And metadata associated with transcription factor datasets are stored in JSON files, and specific transcription factor datasets are fetched from the ENCODE portal. So in terms of a demo, the query interface looks like this. And here on the left, you have the different types of data sets, and here you have the different experiments that have been conducted, and you can upload your private data and submit your query. And so here is the query browser where you input your gene, and then we also have, for every study type, we have a lot of information. So here, for example, we have whole blood from Framingham Heart Study and 5,000 individuals, and you can also select based on ancestry, African, European, Asian, and etc. Whatever is available within the data sets, we provided them, and you can also link out to the specific study that you're interested in. So for example, if we do a simple query for a cis-Eqtl, and for JAK2, we for example, we choose 3 monocyte exposure data sets, and then we chose public data sets, and we just submit our query for JAK2, which displays the results for cis-Eqtl. And we have the tracks for the cis-Eqtls within the IGV browser, and then every Eqtl is a red dot that you see. And if it's only within one study and it's not shared among different studies, it's in blue, and if it's not, it's in red here. These are all shared. These Eqtls are all seen in multiple different study types. The GWAS catalog is here, and then the different isoforms of the genus here, and here you have the legend of the information. You can zoom in and out and like look at the different regions of the gene. And for example, you can click on the cis-Eqtl and get its p-value and other associated information, and then you can click on the GWAS variant and again get all the associated information. And similarly, you can query for trans-Eqtls. Here we query for a set of genes, and then you get a p-value based ordered plot. Here, this is a trans-Eqtl that has the highest p-value, and I want to point out that this is arguably the only interface for querying for trans-Eqtls. And from hovering these data sets, you can get p-value and all the other associated information. And then if you click on it, then you get more information, and you can, query. So for example, you may want to know which other genes does this trans-Eqtl alter, for example, what are the other targets and all the other information, different databases it's also linked to. And you can also do a Manhattan plot and look at the chromosome based on the chromosome level. So here you can see the highest p-value, which chromosome that it is, associated with the genes that you're interested in. And for transcription factors, again, you can just input the transcription factor interested, and after you query, you can find the transcription factors that are known to modulate the gene that you're interested in, and you can look at the detail. You can click on which study that you want to look at in detail, and again, it opens an IGV type browser. So ImmuneRegulation so far has about, for the last 6 months, it has 3,500 visitors from 43 different countries. Well, we developed it in US, so I guess most users are from the US, but it's also around the world. Most people are accessing ImmuneRegulation through their desktops, and some of them through their mobile devices, even though we developed it mainly for desktops. And they usually look at it directly, and some go through social media, etc. So in conclusion, ImmuneRegulation provides the largest database of immune regulation data, interactive interface to visualize and discover associations between genes and regulators, and it enables immune researchers to explore regulators of gene expression to explain cohort or cell type dependent gene expression variations, select ideal cohorts or cells for follow-up studies, develop molecular signatures of immune response, or facilitate their future application in patient management. So all these different types of applications are some of the things that would be useful for immune regulation. The website, I put it in multiple slides, but you can access through this website. We just published immune regulation and nucleic acid research, so you can download it here. We have a poster, B14, today at the poster session of BioVis. And you can also follow us on Twitter. We have ImmuneRegulate handle, and we love feedback. Please feel free to email us, either me or the lead author, Selim Kalaycı, who is a postdoc in my lab. And overall, this is a collaboration of multiple investigators because it originated from the Immune Project Consortium, and these are the individuals that provided us additional data that is only available within our interface. And these are the investigators that are associated with the development of the tool. So thank you very much, and I'll be happy to answer any questions you may have.
So do we have a question? Meanwhile, I would like all of you who are presenting a fast forward to gather at the front, ready to jump in onto the stage when your presentation appears.
All right.
Any questions? No? I have one question.
Yes.
So, did you have to work with the IGV team or IGV.js to enhance it?
Yes, there were some issues with incorporating IGV. So for those issues, we communicated with them, but we didn't have any extra additional requirement or request. It was just to be able to embed IGV because, of course, whenever you implement something, there are some errors, and they were extremely helpful in resolving them. Thank you.